My name's deGraaf, and in this presentation we're going to start looking into the reproductive physiology of rams. So specifically, I want to talk about the production of functional sperm, spermatogenesis, or the genesis of sperm, and also epididymal maturation. It's something which is almost always glossed over, that sperm need to have an additional period of maturation through another structure which sits just next to the testis in order for them to become fertile. So let's jump straight on into it. So here you can see the testes. Now these are not the testes of a ram, but of their closely related ruminant partner, the bull. And you can see that this is a fairly healthy pair. Now these testes have two main functions. They produce and transmit male genes, this is via sperm, and they also produce reproductive hormones, which in the case of the testes are generally the androgens, although they do produce a few others as well. Now within the testes is something called seminiferous tubules, and it's within these that all of the action happens. So here you've got a cross section through a pair of bull's testes. You've got this pink or orangey kind of area, which is known as the testicular parenchyma. This is all seminiferous tubules. In the middle here, you've got something called the mediastinum, and this is where all of the seminiferous tubules are draining into before they make their way to the epididymis. This is just a stylized diagram of inside of the testes. Now, it doesn't come out like this, but this is the seminiferous tubule, and you'll see that both ends of the seminiferous tubule come back here to the ready testis, which is what the mediastinum actually is, or the structure that the mediastinum actually is. Now this testicular parenchyma consists of two discrete compartments. You've got within the seminiferous tubules, and it's here where there is something called the Sertoli cells, which are like a nurse cell that help the sperm to develop. And then you've got the sperm themselves, which are developing, and you've got sperm in all different states of development. Then you've got between the seminiferous tubules. So here is where the androgens are synthesized. So if you're wondering where testosterone is made, it's made here between the seminiferous tubules in a cell type called the Leydig cell. Now, this diagram here over on the right, we've just got some labelled parts. So interstitial cells, this is basically where all the Leydig cells are. So this is an area which is between the seminiferous tubules, and these structures here, these are the seminiferous tubules, and they're all different shapes and sizes because as the seminiferous tubule is packed into, the seminiferous tubules are packed into the testes, and they're sort of all looped all over each other, then if you make a slice through the tubule, you're never going to get a perfect section, or, well, you will sometimes, but if you go right through, you're going to get a whole lot of different sections of the tubules. So some might be perfectly round, but a whole lot of them are going to be these funny sort of oval shapes here. If we zoom in a little bit closer, again, we can see here this area between the seminiferous tubules, and then here is around an edge. This is inside of a seminiferous tubule. So we've got the lumen of the seminiferous tubule here, and then along here is the basement membrane of a seminiferous tubule. And these dark purple spots, they're developing sperm. They're all different stages of development, from spermatogonia, which is the most immature, right at the base, to the spermatocytes, and then up to the spermatids, which are just around the lumen here. So it's the spermatids, the last stage, and an elongated spermatid is the most mature sperm type before the actually released by the Sertoli cell at a point called spermiation, and it's here where they go from being called elongated spermatids to when they are dissociated from the Sertoli cell, and that's when you start calling them sperm. Now here is an example of if you had a perfect section through a seminiferous tubule. So around the edge is our basement membrane, and then in the middle is our tubular lumen, and all these dark patches are developing sperm. So spermatogonia down towards the bottom, and then at the top you've got the spermatids. Now you can see all these little wavy like structures here, sort of looks like hair. These are actually the sperm tails. So these are elongated spermatids just before they're released by the Sertoli cells into the lumen to drift down towards the epididymis, where they'll undergo further maturation. Now, it's difficult to make out because we're not experts at histology, but down along here you'll also see the nuclei for the Sertoli cells. And the Sertoli cells actually are a very strangely shaped cell that stretch all the way from the basement membrane up to the lumen, and they support these sperm, these developing sperm, as they go through their different stages. Here is an example of the nucleus of a Sertoli cell, and then here you've got a variety of different developing sperm. These here are elongated spermatids, and their tails will be stretching up into the lumen at the top. This is just a stylized diagram of what we've already seen. So you can imagine that along the bottom here, this is sort of as if the seminiferous tubule was curving around here. So that's the basement membrane, that's the edge of the seminiferous tubule. And then we have our Sertoli cell. Now, one Sertoli cell, I'll just trace it with my pointer. One Sertoli cell is stretching all the way up to the top, and it's wrapped around all these different developing spermatogonia, spermatocytes, up the top here, spermatids. That's just one Sertoli cell. You can see that's its nucleus. You can also see that there are junctions between the different Sertoli cells. So here's another junction just here. These junctions are really important because they form what's called the blood-testis barrier. What the blood-testis barrier is doing is it, as its name would suggest, is a barrier for blood. Now, that's important because within blood there are immune cells. Now, in this basement compartment, in the spot between the blood-testis barrier, these are tight gap junctions here, between there and the basal and the basement membrane, this is the basal compartment, and here blood can get in, immune cells can get in, and that's fine because spermatogonia, the initial stages of spermatogenesis, all of those cells are genetically the same as every other cell in the body. So the immune system recognizes it as self. But once you get past the blood-testis barrier, you have these spermatocytes. And spermatocytes have undergone a process, or started to undergo a process called meiosis. And meiosis has a stage within it called crossing over, and it's here where genetic information is exchanged and the cells become different. This is how genetic diversity is introduced. This is why not every offspring, baby or lamb or whatever the species we're talking about, looks the same from the same male. It's because each sperm has a slightly different genetic complement because of the crossing over which has occurred during meiosis. So whilst this is great for genetic diversity, it does mean that if an immune cell was able to get in here, it would go, right, this is not something that should be here, is different to what I've been programmed to identify as my body that I'm protecting, so I'm going to attack it. And the same goes for the spermatids. So if that blood-testis barrier wasn't there, you would have all of these sperm cells being wiped out, and that is what can occur when this blood testis barrier is broken down either by mechanical damage or disease. Blood gets in, immune cells get in, and you get a whole bunch of sperm cells being attacked and eaten by the immune system, and as a result you have low sperm count in the subsequent ejaculates. Now the Sertoli cell, as I've inferred, is a pretty important cell. If there were no Sertoli cells, there'd be no developing sperm, because they physically anchor the sperm to themselves as they're going about their development. They deliver proteins, they manufacture hormones, they remove genetic, they remove cytoplasmic material as sperm are going through the latter stages of spermatogenesis when they're being repackaged from a round cell into an elongated cell. So they're a really, really important part of the spermatogenesis story. As I've already mentioned, they're all linked to each other by these gap junctions, and they help to control the level of hormones which each sperm are exposed to. Now, here is a representation of the shape of each of these Sertoli cells. So down here is the edge, the basement membrane of the seminiferous tubule. Up the top is the tubular lumen, and you can see all these funny little cave-like eaten out parts to the cell. Now these would normally be filled with developing sperm. So down the bottom you're going to have spermatogonia. Up in the middle section you're going to have spermatocytes, and then up the top is where you'll have your spermatids. Now spermatogenesis, as I've already stated, is the genesis of sperm. It's forming spermatozoa, and it consists of three phases. Mitotic proliferation, which is just lots and lots of mitosis, making lots and lots of sperm. There's meiotic division, so this is where the different genetics are brought in, where there's crossing over, so there is genetic diversity introduced. And then the third phase is got two different names depending on which textbook you read. It's either called cytodifferentiation or it's called spermiogenesis. And this whole process of spermatogenesis is just constantly happening inside of the testes. From the time of puberty onwards, the testes are always producing sperm, and depending on the species, it's something in the vicinity of 300 to 600 sperm per gram of testes produced every single second. That is a lot of genetic material being produced by those gonads. So let's take a look in a bit more detail on the different stages of spermatogenesis. So here we need to flip how we've been thinking about it so far, where we had the basement membrane down the bottom and we had the tubular lumen up the top. Here it's the reverse. So up the top we've got our basement membrane. So you think of this as being inside of a seminiferous tubule. We've got our basement membrane up the top, and then as we work down towards the bottom, we're going to get closer to the tubular lumen. So here we have mitotic proliferation happening. We've got spermatogonia dividing. Depending on the species, different number of divisions. It's not important. The most important bit is just for you to know that lots of copies are made. These spermatogonia just proliferate. And this all occurs in the basal compartment of the tubule, where you don't need an immunoprivileged site. So this is before we get to that blood testis barrier. Now, it's not something that I'm going to get into in this presentation, but you might be thinking, well, is there a constant supply of spermatogonia? Wouldn't they run out? And it's not well understood, but it appears as though some of the A4 spermatogonia turn back into A1 spermatogonia, and that allows there to be this constant regeneration. Now this second phase, meiotic division, generation of genetic diversity, the number of chromosomes in the cell halves, and here the developing sperm cells are called spermatocytes. To begin with, they're called primary spermatocytes, then they're called secondary spermatocytes, and this is occurring in the adluminal space. So this is the point from the blood testis barrier up towards the lumen. And then the last stage we have is cytodifferentiation or spermatogenesis. Now this is where the genes are packaged for delivery to the egg, and sperm go from being these round cells which can't do a thing to being the elongated spermatids with a tail that can propel themselves to where they need to go. See these grayed out bits? This references the fact that the entire process isn't perfect. It is a process which can be interrupted by something called apoptosis. So if there's something that goes wrong for whatever reason, you can have cells just not developing on. And if it occurs, the earlier it occurs in the process, the more attrition there is to the overall sperm number from an initial A1 spermatogonia. But it can happen anywhere in the process, and if it happens in at large enough levels, then you will get lower than expected numbers of sperm in the ejaculate. And we'll get on to what it means to adjust the levels of apoptosis or the efficiency of spermatogenesis and how that can change the overall sperm output that you might see in an animal. This just shows the several stages that occur within spermatogenesis. I'm not going to go into them in any kind of detail. I just want you to be aware of the fact that this is what is occurring right there up towards the lumen when the cell is being repackaged, and there's a whole bunch of cytoplasm. This is the stuff inside of the cell which is being removed by the Sertoli cell. So again, very, very important cell. Now, sperm come in all different shapes and sizes. Some of them are really weird. In agriculturally relevant species, you can see that they're around about 60 to 70 micrometers, and their head shape, rather than being round or sort of oval shaped, which is the case in humans, sperm are more of ram sperm, bull sperm, boar sperm, horse sperm. They're more of a paddle shape. So what I mean by that is that if you look at them here by this view, that they look like they're a nice oval, but if we rotated them through 90 degrees, we would just see a very, very thin line. So they're just like a paddle. See a little bit more of an idea here about what that would mean. Now, inside of the sperm head, we have all of the DNA. So this is the genes which have been packaged for delivery. On the outside of the head, we have something called the acrosome. The acrosome is really important because inside of the acrosome are the enzymes which are necessary for the sperm to get through the egg. If you don't have an acrosome when you turn up to the egg, then you are not getting inside. So very important, and something that we can test for when we do sperm testing. And here we have the tail. It's made up of a whole bunch of different bits and pieces that you can go away and read about if you're really interested. There are no muscles in there, but there are mitochondria which pull microtubules up and down and cause the sperm tail to move. The overall rate of spermatogenesis is constant. Now, what that means is that the amount of time that it takes for sperm to go from being A1 spermatogonia all the way through to elongated spermatids being released by the Sertoli cell is fixed. Now, there's a lot of information on this slide which I don't want to get into in too much detail, but essentially what is occurring through the process of spermatogenesis is that there are a number of different levels which the developing sperm have to go through, and there's 4.5 levels that the sperm cells go to. Imagine it's kind of like an elevator, and each cycle the cells are getting more mature and they're moving up a little bit further on this elevator, and it's taking 47 days to get from the basement all the way up to the top floor, the lumen, and to be released. But you might think that if this is just occurring in like a cyclical process, then wouldn't that mean that there are just pulses of sperm which are outputted by the male? Why is it that on any given day you can expect that there should be around about the same number of sperm in an ejaculate? Or the other way of asking this question is, how is a constant supply of sperm maintained? And the answer is something called the spermatogenic wave. So this refers to the fact that at different points along the seminiferous tubule, so imagine that this is a section of seminiferous tubule which is running along, and this will keep going for quite a distance, and there's a lot of them inside of the testes. If you take a slice through any given part, you will see sperm developing at any one of 8 different stages. These are just fairly arbitrary cellular associations, but if you look at this little section through here, it's placed at stage 1. So what that means is there are just round spermatids here at the top. And there's a bunch of other cells underneath, because remember they're going up on the lift. But if you go a little bit further down the seminiferous tubule, take another slice through, you can see that here's some cells which they're just a little bit further along, and they are going to be released in a few more days. But if you then, in this same seminiferous tubule, went another few centimeters along, you might find this lot. So this is stage 8, and this shows cells that have matured right to the point of being released. So these mature spermatids are just about to be released, and when they are released, then this particular slice will go back to being a stage 1. You can see that the round spermatids underneath are what you would see here. So like I said, it's like a lift or a conveyor belt, something along those lines, if you want to think about it in those terms, where you've got constantly developing sperm coming from the basement up to the top. And because the seminiferous tubules are so long, because every slice you can find sperm at a different stage of development, you've always got sperm being released. There are always sperm being released into the seminiferous tubule, so there are always sperm heading on down to the epididymis. There are always sperm being added to the reserves ready for ejaculation. Now I want to remind you that just like the female, reproduction in the male is under hormonal control. So we have the HPG axis, which I've gone into in a reasonable amount of detail in some of the other presentations. Here we've got our hypothalamus, we've got our pituitary, and then we've got our gonad, which is here represented by some Sertoli cells with developing sperm inside of them. So this is our testis. So here we have GnRH being released, acting on the pituitary. Pituitary makes LH. LH acts on the Leydig cells to produce testosterone. Testosterone supports spermatogenesis. And then you actually can get estrogen produced from testosterone. That's how it's made. There's an enzyme which converts testosterone to estrogen. And all these things negatively feed back on the axis, just keep everything in tune. Inhibin will negatively feed back on FSH, which is not actually shown in this diagram, but FSH can be involved in regulating spermatogenesis too. And these graphs just show how intimately involved those different hormones are. So time along the bottom, relative blood hormone concentrations up on the y-axis, and you can see that for every pulse of GnRH, you're getting a pulse of LH and FSH. It happens again and again and again, like clockwork. Likewise, at the level of the gonad, got LH that has been produced by the pituitary, and you can see that after every LH pulse, you're getting testosterone being produced. So the whole thing is under hormonal control. So if you want to think about efficiency of spermatogenesis, you got to think about anything that would affect the endocrines, but you would also want to think about any of these other factors that I've got listed here, which could alter the efficiency of how sperm are being made. So another way to think about it is factors that might affect the weight of the testis, because remember that there was 300 to 600 sperm being produced per gram of testis per second. So if you can increase the weight of the testes, then you're going to be making more sperm. Or if you're able to increase the efficiency that sperm produced per unit weight of testis, and that is largely down to the amount of apoptosis which is occurring. So if you can reduce that. So just a little summary there, that the timing or rate of spermatogenesis cannot be changed, but the efficiency of spermatogenesis can. So this rate of sperm production can be altered by altering these factors. Now, epididymal maturation is the next step. So it's not enough that sperm are just produced by the testes. They also have to undergo a next stage of maturation. So here we have our testis again. Now, this I touched on right at the beginning, that after sperm leave the seminiferous tubules, they go into something called the ready testis, or the seen as the mediastinum if you cut the testes open. And this ready testis goes all the way down to the head of the epididymis, and then the epididymal duct goes all the way down through the body of the epididymis to the tail, and then towards the vas deferens and up and out of the body. So when sperm leave the Sertoli cell, they're immotile, and they actually just float down along this seminiferous tubule. And it's not until they get to the efferent ducts, which are just a little bit further along than the ready testis, where there is some cilia and some smooth muscle contraction to push them along. Prior to that, they're really just like they're on one of those tube rides where they're just slowly going along, and there's kind of a pump which is happening because you've got absorption of water down in the ready testis, but you've got fluids being produced in the seminiferous tubules. So it's just like sperm being on a nice relaxing tube ride. Now when they get to the head of the epididymis, there's lots of fluid being resorbed at this point from lots of the fluid that had been produced by the Sertoli cells being resorbed by this structure within the epididymis, by the head. And so as a result, the sperm are getting concentrated more and more, over 100-fold. And inside of the epididymis, there is some muscular contraction, and they are beginning to get pushed through. So in the body of the epididymis, there is a modification of the environment, and as a result, the sperm are exposed to a different environment and begin to mature. And in the chorda, you have storage of the sperm. And this passage, again, depending on the species and depending on the frequency of ejaculation, can take anywhere from 6 to 14 days. Now, a lot changes about a sperm cell, but these are the key things that change when they go through the epididymis. So at the head, or the caput in Latin, sperm are immotile, they're infertile, and they have something called a proximal cytoplasmic droplet, which has actually, unfortunately, gone a little bit down our diagram here, but it should be sitting up more towards the head. So imagine that's up there. But once the sperm have made their way down to the cauda or the tail, these sperm have become progressively motile. They've lost their cytoplasmic droplet, or at worst, it's distal, so it's towards the end here of the tail, and importantly, they're fertile. Here's a list of all the other things that happen to the sperm during their passage through the epididymis. I'm not going to read them out because you can pause the presentation if you'd really like to and read them yourself. But the important thing for you to be aware of is that lots and lots of things are going on here inside of the epididymis. So if you have a dysfunctional epididymis, you can end up with dysfunctional sperm as well. So really important structure. So that brings us to an end in this short introduction to how sperm are produced by the male and how that entire process is regulated. To summarize our lesson from today, I would love you to remember that the testes are there to produce sperm and hormones, and they contain some really important cells called the Sertoli cells and the Leydig cells, and these are there to support the reproductive endocrinology and also the development of the sperm. Spermatogenesis is the creation of sperm by mitosis, meiosis, and then cytodifferentiation, which is that last stage, also referred to as spermiogenesis. We learn about the cycle of spermatogenesis, but we also learn about the spermatogenic wave, and these things put together give us that constant sperm production in the animal. The whole thing requires testosterone and is under control of the HPG axis. The efficiency is influenced by a variety of factors, which I didn't really talk about, but suffice to say that if you have disease or damage to the testes, then it's going to be bad for sperm production. And lastly, we learnt that sperm require further maturation in the epididymis, and I didn't touch on it, there's even further maturation which occurs during ejaculation by the addition of seminal plasma, and then inside of the female reproductive tract as well. They undergo something called capacitation and also the acrosome reaction when they meet up with the egg. That's a good scientific summary. What about an applied summary? So I'd say here, if you're looking for take-homes, if you've been sitting here thinking, gosh, this is far too much detail, I don't need to know all of this, please leave me alone, Simon. Well, what I would tell you to take home from the last 32, 33 minutes is that semen production takes 7 to 8 weeks in the ram. That nutrition and environment can affect semen production by affecting the weight of the testes and the amount of sperm which die during the spermatogenic process. So if you're going to be providing advice to people on changes to ram management, be aware that changes to ram management affect semen 7 to 8 weeks later. It takes a spermatogenic cycle in order for any changes to be reflected in subsequent semen quality. So this is really important when you think about the types of things which are being done to rams prior to a joining or prior to a semen collection. You need to avoid stressful or potentially damaging influences which those rams could be exposed to as they go towards being joined. So to try and get anything that could cause an issue for them, get those things done at least 7 to 8 weeks before you need those rams to be firing. So that would be my applied summary for today.